Hi everyone, in this video we will be discussing about classification of medically important bacteria, and we will be focusing mainly on the classification based on their shape and gram stain. Based on shape of bacteria, they are classified into cocci, that is, they look spherical in shape. Next one is bacilli. These are rod-shaped cells. Next we have is spiral bacteria, and as the name suggests, they are spiral in shape. Then we have curved or comma-shaped bacteria. And finally, we have Coccobacillus. These are bacteria whose shape is a combination of a coccus and a bacillus. There's also a variation in distribution that we can see in these bacteria. It can occur singularly, can occur in pair, hence it's called diplococci. Then there are staphylococcal species that appear in clusters. Then we have streptococcal species that appear in chains. Similarly, bacilli can also appear singularly or as diplococci or in chains as for streptobacilli. Based on gram stain, bacteria can be classified into a gram-positive and a gram-negative bacteria. These bacteria differ from each other in regards to the structure of their cell wall. Gram-positive bacteria, they have a thick peptidoglycan layer, whereas gram-negative bacteria, they have a thin peptidoglycan layer, but it has an outer membrane external to the peptidoglycan layer. As you can see, this outer membrane is absent in the gram-positive bacteria. And this is the basic principle of gram stain. It is very important, and we will revisit this concept again later while going through the steps of gram stain. Now let's look at what is gram stain, and we'll also quickly go through the steps to perform a gram stain. Gram stain is one of the most widely used staining techniques in microbiology. This staining technique was originally devised by Hans Christian Gram in 1884. This technique consists of 4 major steps and 2 different dyes. These dyes impart different colors to different bacteria and hence helps to differentiate the bacteria into a gram-positive and a gram-negative bacteria. Now let's look at the 4 basic steps of gram stain. First of all, we need to add few drops of crystal violet stain, this is the primary stain, to the heat-fixed smear. Then wait for a minute and rinse it with water. Then we add few drops of grams iodine, again wait for a minute and then rinse it with water. Then we add few drops of decolorizer, that is ethanol, and then we wash it with water again. And lastly, you counterstain with safranin. Then wait for a minute and rinse with water. So basically here the 2 main dyes that we have used are crystal violet and safranin, and then we observe it under microscope. The gram-positive bacteria, they retain primary stain, that is the crystal violet, and hence appears violet or purple in color. Whereas the gram-negative bacteria, they are decolorized by ethanol, and hence they take up the counterstain and appears pink in color. Now let's look at the principle of gram stain. In the first step, when we are adding crystal violet to the smear, all bacteria, they take up this dye. Now in the second step, the iodine is used to fix the dye to the cell wall. It forms a crystal violet iodine complex, and hence the crystal violet dye cannot be removed easily. Now in the next step, adding this decolorizer, it tries to wash away the stain. And here is what we need to understand, the concept of cell wall of a gram-positive and a gram-negative bacteria that we discussed earlier. The gram-negative bacteria, it has an outer membrane and it is a lipid layer. So when we add the decolorizer, it dissolves this lipid layer and hence the gram-negative bacteria loses its primary stain and appears colorless at this stage. Whereas the gram-positive bacteria, they have a thick multi-layered peptidoglycan layer with an outer membrane, and hence the bacteria remains stained, that is, it appears purple in color. Now in the final step, the safranin is easily absorbed by the gram-negative bacteria and it appears pink in color. Hence, this difference between the cell wall composition between a gram-positive and gram-negative bacteria is the basic principle of gram staining. And being able to differentiate between these 2 bacteria is clinically important as it provides guidance to empiric treatment while pending for definitive culture. Now that we know the classification of bacteria on the basis of their shape and gram stain, let's look into examples of some of the medically important bacteria. Based on the gram stain, bacteria can be either gram-positive or gram-negative. Let's look into the gram-positive bacteria first. Depending on their shape, they can be either cocci or bacilli. The gram-positive cocci can be further categorized into catalase positive and catalase negative. The catalase positive includes the Staphylococcus species, which can be further categorized into coagulase positive, that is the Staphylococcus aureus, and coagulase negative, that is Staphylococcus saprophyticus and Staphylococcus epidermidis. So now that we have covered staphylococci, let's look into the streptococcal species, which are catalase negative. Now, based on the hemolytic reaction, these streptococcal species can be further differentiated into alpha, beta, and gamma hemolytic strain. The alpha hemolytic strain includes Streptococcus pneumoniae, viridin streptococci. The beta hemolytic strain includes Group A Streptococcus pyogenes and Group B Streptococcus agalactiae. The gamma hemolytic strain includes Enterococcus faecium, Enterococcus faecalis, and Streptococcus bovis. The gram-positive bacilli are categorized into aerobic and anaerobic. The aerobic organisms include Bacillus and Listeria, and the anaerobic organisms include the Clostridium species. Now let's look into the gram-negative bacteria. Based on their shape, they are further categorized into bacilli, cocci, coccobacilli, and curved or comma-shaped bacteria. The gram-negative bacilli can be further divided into groups based on lactose fermentation. Organisms that ferment lactose include E. coli, Klebsiella, Citrobacter, and Serratia. Organisms that do not ferment lactose include Shigella, Salmonella, Proteus, and Pseudomonas. The gram-positive cocci can be further categorized based on maltose utilization into Neisseria meningitidis and Neisseria gonorrhoeae. The coccobacilli includes Haemophilus influenzae, Bordetella pertussis, and Brucella. And finally, we've got the curved bacteria, which includes Campylobacter jejuni, Vibrio cholerae, and Helicobacter pylori.